Today we'll be collecting DNA from two mutant plants, or two varieties of mutant plants, both with a mutation in MKP2. And on the left-hand side here, we have MKP2 with a Salk mutation, and on the right-hand side, we have MKP2 plants that have just a CS number, and we believe that these right-hand side plants are going to be homozygous for the mutation. So I'm going to start by doing 8 DNA preps on these first 8 MKP2 CS plants. I'm using a snippers to clip off a small leaf, maybe this small leaf, from a plant that I've numbered 5. Before I go on to the next plant, I'm going to take the snippers and wipe it on a paper towel and do some snips. This paper towel has got 95% ethanol on it. So I know I'm getting rid of the DNA from plant number 5, and I can go on to collect the leaf from plant number 6. Now that I've collected the leaf from my plant, I can place it into the appropriately labeled tube over here. So I've got tubes 1 through 8, and I think you can see in the bottom of each of these guys, we've got a little piece of leaf. So now I've collected leaf tissue from plants 1 through 8. The next step is to grind up this leaf tissue, and I'm going to use this pestle. It's just a plastic pestle that we can reuse, and I'll try and get the piece of leaf to the side of the tube here and mash it around a little bit. So mash, 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 and I'll do this for all 8 leaves. Now that all 8 of my samples have been ground up using the pestles, I'm going to use some Shorty Buffer to help break apart the plasma membranes on the organelles so we can get out the DNA. Shorty Buffer has about 4 ingredients in it: lithium chloride, tris, EDTA, and a detergent. I'll put the recipe in the description box below. So we're going to add 400 microliters. You can see I've got my pipetter set to 400. We're going to use 400 microliters of Shorty Buffer in each tube. Because I'm being careful not to touch the plant tissue in the tubes, I can use the same pipette tip for all of my samples. Now that the Shorty Buffer is added, we can mix it a little bit further, making sure to mix up any clumps of leaf tissue that are still in the buffer. To move forward with these DNA samples, we next need to spin them down to precipitate any plant material that's left. Alright, I've placed all my tubes into the centrifuge, and you'll notice I've oriented them all so that the little nub of the tip is pointing out, so that we know the orientation of each of the tubes when they come out. I'll spin these tubes now for 5 to 10 minutes at max speed. While the tubes are spinning, I want to set up my next set of tubes that will have 300 microliters of isopropanol in them. I'll also label these tubes. My samples, my original samples, have been spun down, and I've made new tubes that have isopropanol in them. And you may notice I've also labeled these tubes much more thoroughly than my original tubes, because these second tubes with isopropanol will be the ones we'll store and use long term. I've put 300 microliters of isopropanol into each second tube, and I can now pull off the supernatant liquid, about 300 microliters from our original tube, and into the isopropanol. I'll repeat this for all 8 tubes. Now that I have my isopropanol and my plant liquid combined together, we're going to spin this down. We're going to first invert the tubes for a couple 30 seconds, and then we're going to spin these down for another 5 to 10 minutes. Because DNA won't mix with alcohol like isopropanol, as we spin this down, the DNA will be precipitated along the outside of the tube, while the other plant materials and the isopropanol will stay in solution in the middle of the liquid. You can hear my isopropanol plus DNA samples spinning down for about 10 minutes in this centrifuge. With my tubes removed from the centrifuge, I'm going to pour off the liquid because the DNA is captured on the side of the tube. Even if you can't see it, it's there. So I'm first quickly pouring off this isopropanol, and then I'll rinse the DNA pellet with 95% ethanol. The longer we let this pellet sit in the isopropanol, the more likely it is to come off the side of the walls, and so it's best to dump the liquid immediately. Next, I'll rinse these pellets with 70 to 75% ethanol. Because I know the pellet is at the back of the tube, I'm going to aim my pipetter toward the front of the tube, and then I can just pour that liquid all right back off. Because I didn't touch the pellet or any of the liquid in the tube, I can use the same pipette tip and move rapidly through all the samples. Last but not least, I need all of these pellets to dry. The ethanol will dry off very quickly, and so we can just invert the tubes and sort of pat down the liquid, and then we can leave these here either standing up or falling over to dry in the next 10 minutes. They can also dry overnight, but that can make resuspending the DNA a little more difficult. However, my boss in graduate school would always say, "Do you really believe that there's no DNA in there? Would you be willing to take that risk?" And of course I do believe there's DNA in there. We'll resuspend it in just a few minutes. Alright, these samples have been drying for about an hour, so I can invigorate them. I can elute them with water or with TE Buffer, and today we're going to use water. I've got some nuclease-free water. I've got 200 microliters set on my pipetter, and I'll just add the water to each of the samples. And again, because I'm not going to be touching the samples themselves, I can use the same pipette tip all the way through. Depending on how concentrated you want your DNA to be, you can use anywhere from 200 to 500 microliters. Today I'm using 200, and we are going to use this DNA to do PCR to identify whether our plants are carrying the mutation we're interested in. This DNA can now be vortexed to mix the DNA into the liquid and then frozen in the -20 for long-term storage.